Welcome to The Center. This is a podcast from the Center for Neurotechnology and Neurorecovery at Massachusetts General Hospital. In this podcast, we will be interviewing faculty members, clinicians, and researchers at the Center for Neurotechnology and Neurorecovery to learn about their fascinating research and the impact it has on their patients. Today, I'm lucky to be joined by Dr. Leigh Hochberg and Dr. Sydney Cash, who together co-direct the Center for Neurotechnology and Neurorecovery. Thank you both for joining me today. Could we start by having you introduce yourselves and telling us a little bit about your research?
Sure, Lee, why don't you start?
Sure, Josephine, I'll be happy to. So my name's Leigh Hochberg. It's great to be here. And I'm a critical care neurologist and vascular neurologist here at MGH. Most of my research is focused on developing technologies that'll restore communication and mobility for people with neurologic disease or injury. And a good portion of that research is focused on what we refer to as brain gate, which is a trial of a brain-computer interface that I suspect we'll get a chance to chat about over the next hour.
I'm Sid Cash. I'm also at Mass General Hospital, and I'm also a neurologist. I focus primarily on caring for patients who have epilepsy. And in my research, I'm trying to develop techniques to better understand how the brain works and transitions to states like seizures so that we can control it better and alleviate many different neurological diseases.
So what do you think is the most significant, if not most important, advancement that propelled the field of neurotechnology to the point it is today?
Again, no, you can go first.
All right. So most important, it's going to be really hard, but I think I'll kind of take a historical perspective. So even going back to the 1930s and 40s, there's one paper that I read as a graduate student that really stuck out. It's written by Marianne Hines, who can't remember whether she was then or would later become the chair of anatomy at Emory. She made an observation while she was stimulating the top of the brain, that is stimulating the cortex, that if she placed an electrode a little bit deeper into the cortex than just at the top, that if she used a little less power, that is a little less current or voltage, when she was stimulating, she could still get these complex movements of a limb. And that was it's as I remember the paper, it was kind of in passing that she observed that if you could stimulate a smaller group of cells in the brain, you could still, even with fairly low amplitude, as we might describe it, or fairly low current, you could still get this complex movement of a limb. And it was one of the first, to me, at least in my reading, and Sid will probably go back further in a moment, it was one of the first times, at least, that I recognized that historically, people knew that tiny ensembles, tiny groups of cells in the brain were really important, and that maybe even individual neurons in the brain were really important. So that was one. That was kind of dating back to the 1930s or 40s. To kind of put another group of time together through the 60s and 70s, there was a group of scientists, Ed Everts, who began to record from single neurons in the brain and relate those to the movement of a limb. Eb Fetz, who was also recording from single neurons of the brain and allowing those neurons to control an external device, that is a meter. Carl Frank wrote this amazing paper. It's actually a lecture that he gave around 1967 or 68. He was opening up a new lab at what was then called the National Institute of Neurologic Diseases and Blindness, NINDB, what would eventually become NINDS. And Carl made this incredible statement at the beginning of his lecture that his lab was going to be set up so that one could take recordings from the nervous system and allow those recordings to drive any number of devices. And I won't remember the exact quote, but he talked about controlling teleoperators and a few other things. And then the last statement was kind of the drum roll before the last statement in a sense. It was, and even computers, which was a big deal in the in the late 60s, is what could be controlled directly from the nervous system. And then, I think together with Don Humphrey and Ed Schmidt and others, who were some of the first to take these recordings, and then being able to predict what an animal was actually doing from this neuronal ensemble, that is, from these groups of brain cells. That to me was a huge advance, and really set up the field for kind of where it's headed today.
Yeah, though, looking at the history of what was done and what was understood going back, those are really important understandings and discoveries. I have a broader idea of also what's been really essential for the development of neurotechnologies that we see now. And you already mentioned it, and that's computers and our use of computers. We've had this explosion of understanding of what's happening in the nervous system due to all sorts of technologies. Being able to take that and do something with it on a real-time basis has really been only possible because of the kind of computing power that we have now. And really being able to shrink down our ability to analyze the data that we get and do something meaningful with it, I think has had a huge, huge impact on the field in general, and allowed us to advance in ways that we couldn't imagine back then in the 50s and 60s. Well, they can imagine it. They were thinking about it, right? They were planning for it, but we really didn't have the power to do it until fairly recently, when we've now got the computational capabilities to look at what the activities of hundreds of neurons are doing at a time, or many, many channels of recording are doing at a time, and actually do something with that that's actionable, and actually start talking to these devices in the end. The teleoperators are now here. We could actually do something sort of in a meaningful way with it now. So I really think that's an important advance in general, and obviously for society as a whole, but really neurotechnology has had a big impact.
Yeah, I mean, those computers that Carl Frank and others were talking about using in the late 60s, which, in this room would have taken up the wall of this room.
Or whatever. Yeah,
right, exactly. And I remember those punch cards as well. But the computer that we have in our phone, in our pockets, is orders of magnitude more powerful than what they were referring, even at that time, as the computers that might be controlled. So it's been it is really I agree, incredible advance.
And lets us do things we never really could have imagined doing, frankly, without that kind of capability.
Could you tell me a little bit about your journeys into the field of neurotechnology?
Yeah, so I don't think I really joined the field of neurotechnology until after I had finished my medical training, actually, until I was, really well into residency. I'm not even sure how much I even knew of it. I was much more focused on what can we learn about the nervous system? How can we understand the nervous system? So I did undergraduate work with Leigh Hochberg at Yale, where we looked at single neurons and innervation of single muscles in Drosophila. I then did a PhD with Mu-Ming Pu and Raphael Yusti at Columbia, where again we were mostly focusing on individual neurons and how they did computation, how plasticity was evident in individual neurons, and all of this research was done in model systems. So Drosophila, with the case with Leigh Hochberg, and in various in vitro preparations with Mu-Ming Pu and Rafa, and it was pretty removed from sort of the environment of the clinic. And once I got to residency, I thought, hey, I really want to apply some of this to diseases, to patients. And that's when I sort of began to really understand the relationship between understanding neuroscience at that single cell level, or the small groups of cells, and interfacing with technologies in ways that we could help our patients. And that's sort of how I got more and more interested, and began seeing how what I had been doing and what I've been thinking about can actually be leveraged to understand and then actually go on to create systems that could be helpful in the way that neurotechnology can. So it was a different sort of endeavor than I think you did. You were much more sort of deliberate in a way, or knowledgeable, I should say.
I question both deliberate and knowledgeable. There is a linear path, I guess, but the whether it was purposeful and reflects any knowledge, so it remains to be seen. So, much like your first question, I was thinking about the 1930s. My interest in neuroscience, I peg, at least initially, to my grandmother who had Alzheimer's disease, and that's a very common story that you'll hear from a lot of neuroscientists, that there's somebody in their family, there's somebody who they knew who had a neurologic disease or injury, and the same is true for me. And she lived with us for a few years when I was a kid, when I was 10 or 11 or 9 or 10, so, and that was just old enough to know that something was wrong, but not really being able to understand what it was. And I had memories of my grandmother from before then, who reliably would have the dish of Starburst. That's whenever I went into her apartment, and it was always refill. I have no idea how there was always a dish of Starburst there, but the
hard candy Starburst.
yeah, they were great. And then as she began, as I kind of began to understand a little bit at that time, but didn't understand why, I began to have difficulties with her memory. That undoubtedly had an impact on me. And then skip ahead to being an undergrad at Brown. Really loved the introduction to neuroscience course, Neuro 1, as it was known, still known now. And that, and part from that course and prior experience, I was able as I guess the summer between my sophomore and junior year, worked with a neurologist in the Bronx, Howard Crystal at Albert Einstein School of Medicine, and he had this amazing project that he wanted me to do, and which, getting back to this Alzheimer's kind of interaction and experience, he wanted to determine whether people who were having difficulty with their memory from Alzheimer's were better at sometimes during the day with their memory than at other times. So he and I programmed up a memory game on, and I think we programmed it in BASIC. I'll have to think about that. And I would take what was then a big laptop and travel around the Bronx to the apartments and homes of people with Alzheimer's disease. And we'd do it once in the morning and again some other time in the afternoon, and try to compare. And even though that was the focus of the research, what happened over and over again would be the same people who were letting me into their homes, undergrad, to basically play this early video game or memory task, always wanted to play cards. And they wanted to play, poker or gin rummy, something that required some bit of memory. And no matter how impaired their memory may have been on that formal task, every single time they would crush me on whatever card game it was that we were supposed to be playing. So even then, there was something, and I just kept seeing this over and over, that there were some domains of memory that were preserved, and others that were not. And so there was another moment of interest. And then, as as an undergrad, as you were talking about you're at Yale and me and Brown, and there was this amazing kind of wet neurobiology course where we began to record single cells and see them on an oscilloscope, with a little green tracing, as was the way this was done, as you remember as well. And it was amazingly cool. And that led me to start doing some bench research as an undergrad. The story's going on for too long. We have like 6 hours, right? I'll speed it up. That led me in Ford Ebner's lab to begin to do a little bit of research related to learning and memory, and then eventually led me to apply to MD PhD programs. Common theme. And I remember a cold day, getting on a plane and flying to Atlanta. Warm day suddenly, and I met Professor of Physiology Don Humphrey, and we were talking about research. And I asked a question along the lines of, so what do you think your lab is going to be doing over the next few years? And he said, well, I think I'm about to get a contract from the National Institutes of Health to record brain activity and to drive a robot arm from that brain activity. And I looked at him and I said, what? and he said it again, and it kind of clicked that was not only a really cool idea, but the immediate utility of that in what at that point was already going to be part of the neural prosthesis program that had existed at NIH since the 1970s. It was clear that technology, if what he said wasn't completely made up, that could become the basis for technology that would help people with neurologic injury or disease. So long answer, I guess, but that was how I got started thinking about this field.
Interesting.
Dr. Cash, you mentioned that your early work focused on animal models and computational approaches to neuroscience. At what point did you really feel that clinical impact of your work?
Yeah, I don't think it was really until I had set up my own lab, had been doing research, and was working primarily with patients with medically intractable epilepsy who are undergoing surgery to try and control their seizures, that I was able to start talking to those patients about what we were doing and why we were doing it and the ways that we were, understanding their seizures better and better. And we still have a lot of work to do, but we have a better idea now of how their seizures start and stop and spread. And without even necessarily having an impact on their care per se, the fact that I was starting to be able to have conversations with them about, hey, we're starting to understand this a little more, and we're starting to come up with new ideas of how we might be able to treat it. I think that's the first time that I started to really get that feedback of, I'm starting to get to a point where I could start affecting their disease in a real meaningful way. So it was, fairly early on, because we were already getting a lot more information than we had ever gotten before. And it wasn't until, much later that we're actually starting to have an effect directly on their care. But already just being able to say to people, hey, we think we know a little bit more about what's happening to you, that already was having an impact, I think. For you, Leigh, was that?
Yeah, no, I was thinking about those interactions that you're having, in the clinic, in the hospital, with your patients who are coming to you with medically refractory, that is, seizures that medicine are not treating adequately, and you're not only being able to provide additional medical care and think about the next steps of their clinical care, but that they're contributing their time to inform the knowledge that'll help other people with epilepsy.
I mean, we started off, we were sort of skipping around a little bit, but you said, what's been the biggest thing in the field? The biggest thing in the field really is the fact that there are patients of all sorts of stripes who are willing to participate in the kinds of things that we do. Nothing that we get done. I mean, there's all these animal models, and they're great and they're very important and powerful, but when it comes down to it, it is the contribution of the patients who are willing to participate in some of this research at different stages that really lets anything move forward. Otherwise, we're just stuck and dead in the water, really.
Yeah, no, that's it. And as a result, I will talk about one of those participants in our clinical study, because I couldn't agree more. In our BrainGate research, we recruit people with spinal cord injury or brainstem stroke or ALS, that is Lou Gehrig's disease, or amyotrophic lateral sclerosis, to have recording electrodes placed in the brain. And if everything works in this implantable brain-computer interface field, as it now is, but at the time, if everything worked the way that we hoped that it would, somebody would think about the movement of their own hand, for example, and be able to control a cursor on a computer screen or something else. And circa 2011, one of the amazing participants in our study, known as S3, she'd been in this trial for a few years. She had controlled cursors on a computer screen. She had typed by kind of thinking about moving her hand to point and click. And we asked her what she wanted to do with this investigational technology. And she said she wanted to take a drink. We said, okay. And we clarified what she wanted to drink. And it was a cinnamon latte. So we said, okay, let's see if we can make this happen. So she was living in an amazing assisted care facility, the Boston Home, in Dorchester, not too far from here. And she was sitting in her wheelchair thinking about the movement of her own hand. We were decoding that neural activity, and what she was controlling was a robot arm. And she would think about moving her own hand. That robot arm would reach out, and in this case, she was able to pick up a thermos and bring it to her mouth and tip it towards her so the straw would go into her mouth. And then a handful of seconds later, she took that sip. And what I remember the moment was I remember her smiling. Yeah. I just remembered this incredible smile and response to being able to take that sip. And then she thought about moving her arm back to the table, and she put the thermos back down on the table. And and clearly that wasn't the first time that she had had something to drink in nearly 15 years. Um, but it was the first time that she was able to do so solely of her own volition. And I'm just seeing that smile, seeing her response, and being so happy about being able to do that. It just reinforced exactly what you just said, which is the amazing participants who join us in any of these clinical trials. They are the reason that this field makes progress, that we learn what works,
Absolutely.
that we learn what almost works, but we still need to make better. And then they provide insight to us. They tell us what's working well, and they give us their own real-time experience of using any of these neurotechnologies, which is just extraordinarily valuable.
Right. Totally agree with that.
I know the scope of neurotechnologies is pretty broad, and you both mentioned several neurologic diseases that are being targeted by neurotechnologies. Could you speak a little bit about the patient populations that stand to benefit from neurotechnologies being used in clinical settings?
Yeah, again, you're right. It's very broadly defined, and you could start with where are neurotechnologies being used right now, and there are various ones depending again how you define it. There are tens of thousands of patients with Parkinson's disease who have deep brain stimulators implanted and have for many, many years, and that's a form of neurotechnology for certain, and many things like that, and other movement disorders as well. So if you want to think about that as a neurotechnology, that's a major patient population that's been well versed in this technological advances. Where is it going? What are the other patient populations? We're talking about patients with severe neurological injuries, talking about patients with epilepsy, for whom there's also neurotechnology that's currently available in the form of brain stimulators of various types. I think that to some extent, the challenging question is which patients is it going to really be ideal for? Which are the patients who it's going to be best for, because it's such a broad range of patients with neurological diseases, psychiatric diseases, really anything where the brain is injured in some way. I think we're going to be seeing neurotechnology used for that.
Yeah, I agree. I think and you've mentioned two groups who are benefiting already. Yeah. People with movement disorders using deep brain stimulators, people with epilepsy using responsive neural stimulation as a technology, people with chronic pain who are using spinal cord stimulators. Right. Yep. These are three of a handful of clinically cleared or approved neurotechnologies that are already in clinical practice, and they can be incredibly effective. I think one of the things that draws us both. All right, I guess I'll only speak to myself. You can decide whether you agree. One of the things that draws me, at least to neurotechnology, is that some of these devices, once placed and turned on, so to speak, can have an immediate impact. Right. And whether immediate is seconds or minutes or hours or even a month or two, that is often much faster than some of the other approaches that we currently have available to us for the treatment of neurologic disease or injury. So there's something about a device that is appealing from the standpoint of its potential contribution to restoring function for whatever that function may be. There are lots of other, unfortunately, diseases and injuries of the nervous system for which I think neurotechnology is going to be extremely helpful. And if we make a big jump, particularly about brain-computer interfaces, restoring communication for somebody with ALS who's unable to speak and unable to write, or somebody with brainstem stroke who may similarly have severe speech and motor and motor impairment. Somebody with spinal cord injury who may be able to speak but may have tetraplegia, that is the inability to move arms and legs. I think all of those the people who have those diseases or injuries, I think, can look to neurotechnology as being one of the sources of potential hope, as approaches that will reduce disability, will improve function, will extend autonomy. I think there's lots of great opportunities here, and I know you've thought about this as well. In some ways, some of the diseases and injuries that I just mentioned are very easy to describe. There's a signal that there's in the brain. Yep. It's supposed to go somewhere else in the nervous system, and it can't because something's been disconnected. There are more complex disorders, which fall into the realm of neuropsychiatric disease or mental health disorders. And one wonders and hopes that these types of neurotechnology approaches, which are already beginning to have some impact, will really have a substantial impact on helping people with mental health disorders.
Yeah, no, I think that's really sort of one of the very many interesting aspects of neurotechnology. We tend to think of it as what's happening now, at least I do, as sort of an early stage, right? We're dealing with the fairly clear-cut aspects of nervous system activity. Motor cortex we understand pretty well because of all the people you've talked about doing great work. We understand that. We sort of understand visual cortex. We can understand how to interact with them. But now we're starting getting to the phase where not only can we start doing that, but we can start thinking about how do we interact with complex network activity of the brain that leads to emotional regulation and anxiety regulation and all these other kinds of facets that's part of neuropsychiatric disorders and other disorders as well. And we're heading in that direction for sure. It's a thorny problem, but one that I think the technology and the researchers are certainly capable of attracting and getting to now.
Yeah, so what's great about CNTR, the Center for Neurotechnology and Neurorecovery, is that the majority of the disorders and diseases of the nervous system that you have brought up are actually being looked at by our various labs. And together you direct CNTR. So I was wondering if you could speak a little bit about what brought CNTR together. What were your motivations to start the Center for Neurotechnology and Neurorecovery, and what are some of your goals?
Yeah. You want to go ahead and talk about goals and start with, and then go ahead.
Sure. Well, so I think the two of us date this conversation to about 2006.
Yes, about right.
And it was actually a great discussion that we had with Joe Martin, former dean of medical school at Harvard, when we were both just a few years out of our residencies and fellowships, and recognizing the incredible impact that a neurotechnologic approach could have to restoring communication, restoring mobility, reducing the clinical impact of seizures, and helping people with other neurologic diseases and injuries. And it was such a small slice of neurologic research at that point. Mm-hmm. The using medicine, that is drug-based research, was well appreciated and well practiced, and a lot of good progress was being made, continues to be made. But using devices to treat or manage these diseases and injuries was really nascent at best. It was, and that discussion, which I remember in his office, yep. Joe was incredibly encouraging of us thinking about putting together a center that would pull from not only the clinical resources that we have, but the scientific resources, talking even then about the really interesting and important ethical components of recording and modulating the nervous system, trying to think about how we were going to train the next generation of not just neurology residents, but of neuroscientists and neuroengineers. Yeah. What do we all need to know in order to move this field forward and actually get to the point that we're creating devices, that we're creating systems that would help people with neurologic disease and injury? So at least when I think about our current CNTR, I think repeatedly about those conversations that we had back.
And part of that conversation and part of that motivation and part of the goals was this underlying concept that we've sort of touched on in the last few minutes, which is that neurotechnology is this incredibly multidisciplinary approach, right? It has engineering in it, it has hardcore neuroscience in it, it has computational science in it, it has the relationships with the patients, it has ethics in it, it has all these different features in order to make it happen. And that's very hard for any one lab to be expert in it, if not impossible. And so we recognized early that, and part of this came about because Leigh was so involved in brain gate and recording from motor cortex. I was looking at patients primarily with epilepsy, but I was trying to get into single neuron recordings in those patients. There was some overlap in the technology and the platform. I needed his expertise to pull that off, and we were doing some computational work. We needed expertise of that, and so we were thinking, how do we actually really make this sort of scale up in a way? And that's where that first conversation, I think, partially came from, and where we've sort of grown this center together to try and get people together, all of whom might be tackling a different neurological problem or some overlapping problems, but to be able to give them the resources and the platform to discuss with each other common problems and solve them better. And how do we teach the next group of people who come through who, need to be understanding the biology and the engineering and so on in ways that we unfortunately never really got. Again, speaking for myself, a lot of it has been seat of my pants, sort of learning it as I go. I wish I had the kind of training that we've tried to make available, or the experiences that we make available to the next set of people who are coming through and making research happen.
Yeah. And even starting our neurorecovery fellowship here was one of the examples of that. Is the residents in neurology, medical students. First of all, we want the best of the medical students to think about neurology and think about it first. Is that a blatant advertisement?
Yeah, no, it's okay.
Fine. So come on into neurology. And then even the fairly early on in one's neurology residency, you begin to think about subspecializing. Are you going to be are you going to see patients in a general neurology practice? Are you going to do a fellowship in a specific realm of neurology? Is it going to be in movement disorders or neurointensive care, or stroke, or epilepsy, or neuromuscular disorders, or any number of other components of neurology? But there's only a very small number who are really thinking about neurorehabilitation early on.
What happens after?
And it's so important to have people who are trained in neurology really thinking about neurorehabilitation, about neurorestoration, about using scientifically rigorous ways to determine which of these methods that are emerging might actually help people with neurologic disease or injury. And as a result, part of what our center has been doing, that is part of what CNTR has been focused on, is trying to really think about the education of residents and fellows and neurorecovery fellows, that so that their careers are focused not only on the acute treatment or on the chronic management of neurologic disease, but really focused on how do we get somebody better, and how do we allow them to return to the lives, to the function that they had prior to the onset of the neurologic disease or injury. Yeah.
Exactly. Yeah.
So for my final question, I did want to ask about the biological and technological hurdles that you feel like need to be overcome to get to a point where you'd be happy with the future of neurotechnology.
I'm already happy with the future of neurotechnology.
There we go. So we agree. That was good.
Well, on the one hand, I'm happy with it. I think we've got a lot of great tools. On the other hand, I'm never totally happy because there's so much we're not yet able to do. Obviously, that's why we're doing this. I think there's a number of technical issues that we're certainly trying to overcome, and we're making strides there. We always are looking for ways to get more recording from the brain. Can we get more sites at higher resolution spatially? Can we keep really good temporal resolution? And really importantly, can we make these things totally transparent, if you will, to the patient, right? Are they going to be implanted in a way that's relatively low risk? Can they be implanted in a way which is cosmetically totally acceptable? Can they be done in a way that's financially reasonable so that they're available to everybody, and not just those patients who have substantial means and happen to live next to Mass General Hospital, but all over the country and all over the planet, can they get access to these? So there's a lot that needs to be done to make it really, really scalable. But there's a lot that's being done progress-wise that's going to happen that way. I don't think that there's any single hurdle that's like an insurmountable problem at this point. Do you think there's a thing that is a big block, or it's lots of little pieces that need to fall into place over time?
I think it's lots of little pieces. And I also, you just touched on this, it's the fact that right now all of these neurotechnologies that we're talking about that are clinically cleared or approved, they record from dozens or a few hundred of the something like 80 billion neurons, at least that's what the textbooks tell us, the 80 or 100 billion neurons that are in the human brain. And similarly, even by stimulating, we're stimulating this particular place over here, maybe also that particular place over there. It's such early days, and yet already the technologies are showing so much promise. So I agree completely. If we could record more for decades, more neurons, more parts of the brain, attending it every moment to, of course, the sine qua non, which is these technologies need to be demonstrated to be safe and then also effective. If we could do that, there's so much good that could come from these technologies.
The other thing is sort of like, to me, the technology isn't the problem. Part of that is just sort of faith in our engineering brethren who are just making more and more and better versions of the things that we need. I think that the onus is going to be on us neuroscientists, because our understanding of the brain and what it's doing and what it's supposed to be doing, I think there's a lot to be done there, right? And to really, like you say, we can record more and more, and that's going to really help us. And now, what are we going to do with this information, and how are we going to use it, and how are we going to, take understanding from that, like the original, FETS papers of what does a single neuron do? Well, now what do hundreds and thousands of neurons do, and how do they interact? How can we interact with them to restore function? That's going to be a real challenge, but I think it's a challenge we're up for as a community.
Yeah, it's a challenge, and it's also a place of friendly tension, for lack of a better word, in the field, that, just as you described, we're both card-carrying neuroscientists, I guess, for lack of a better phrase. Is that a thing?
If there was such
if it is,
agency,
I have my SFN card. I actually do. The which is the Society for Neuroscience. I've got I think I print out my card. And so we think at a first pass about hypothesis-driven science. We have a proposal, we have a thought of how some group of neurons or how the brain may respond in a certain condition, and we really try to do a rigorous, well-controlled, experiment, for lack of a better word, that tests that hypothesis and either gets supported or rejected, and we learn something about the nervous system along the way. And along comes those computers you were talking about before, and machine learning, and artificial intelligence, broadly speaking, which seems to be achieving or helping us to achieve some of our goals for clinical care, for restoring function, for reducing the impact of diseases or injuries. And yet we don't have great insight as to what that black box is doing. And so, the neuroscientist is happy to see the results and a little frustrated that the so-called black box was actually able to get there. But on the other hand, we're beginning to use those tools to get better insights into the nervous system.
Right. That's right. I think that we're learning more and more about how to take those tools, which are incredibly powerful and having a huge impact, now more than ever, and starting to use those for not just getting to a goal, but getting to understanding, which we could then use to get to a different goal or the same goal better. I think that's definitely happening for sure.
Yeah, and just as you said before, it's the people who enroll on the clinical trials in this often hospital-based research that are not only helping other people with neurologic disease or injury by enrolling in the trials, but they're giving us insights into human brain function that we wouldn't have any other way. It's the only chance to get some of this information. And as you said, the people who join us in these trials deserve extraordinary credit, because we couldn't do it without them. We couldn't do it without their insights. We couldn't do it without their time. Sometimes we couldn't do it without them accepting some degree of well-discussed risk. Their generosity there is just amazing.
Well, thank you both for joining me today for our first episode of the Center. And I'm very excited to learn more about your respective research fields in our future episodes.
Thank you.
Thank you.
You too.
Enjoyable.